recording has started for this, and it will be available after we are done here for today, and it will be available through the AAPS website via the PDPD community. I will turn it over to Subrachala, who will be presenting and introducing our speaker for today. So Subrachala, I will pass it over to you.
Thanks, Bo. Hello, everyone. Good morning, good afternoon, or good evening to everyone, wherever you are today. I'm Suvarchala Abari. I'm scientist 2 from Simulations Plus, and I'll be one of the moderator for this webinar, which is organized by PDPD community. So to talk briefly about this community focuses mainly on essential collaboration of scientific disciplines across drug discovery and early development, encompassing novel target and ligand exploration, property optimization, lead candidate selection, and effective transitioning of preclinical species or preclinical candidates into clinical assessment. So today's live event is titled Transitioning from Academia to Industry, mainly a career advice for graduate and the postdoctoral students. And the presenter for today is Dr. Deanna Mudie. She's a senior principal scientist from Simulations Plus. Um, in the physiologically based pharmacokinetic solutions R&D department. She earned her BSc in chemical engineering and her PhD in pharmaceutical sciences from the University of Michigan. And she has over 20 years of experience in the pharmaceutical field. She most recently served as a technical program lead in R&D at Lonza Small Molecules, where she was working on developing new technologies to enable successful bioavailability enhancement of poorly soluble drugs. Then during her doctoral and the postdoctoral work, she worked on developing mechanistic in vitro and in vivo transport models to predict the oral dosage form dissolution and the intestinal absorption. She began her career as an engineer at Merck and Pfizer, characterizing, developing, and manufacturing oral solid dosage forms from preclinical to commercial scales. With that, I'm happy to now present Dr. Deanna Mudie. Deanna, the floor is all yours.
Great. Thank you so much, Suvarchala, for the kind introduction and also for giving me the opportunity to speak. Definitely excited to share my story and then to get questions from the participants as well. So in terms of what I'll be covering for this session, I'm going to share my professional journey and also provide career advice. And then what I'm hoping you'll gain are some tips for success in entering a desired career in the pharma industry, more ideas about how to evaluate industry roles, and then also some insights into contract development and manufacturing organizations like Lonza, where I had previously worked, compared to large pharma roles. So I won't go over my biography again. Thank you, Suvarchala. The one thing I will highlight is my email address here, and I'll be showing that again at the end of the talk, and I'd be more than happy to answer any questions you might have after the webinar. So my introduction into the pharma field first started with my chemical engineering degree at the University of Michigan. During that time, I was fortunate enough that the University of Michigan had a career fair, and I was able to meet some representatives from Merck and Company and get a couple of summer internships. So those internships did start during my bachelor's degree, but I've interacted with and known many, many graduate students that have also done summer internships. So as I'll highlight, this is super valuable and really great for marketing yourself when you're going into an industry field, is having the summer internship experience. So my first one was in Somerset, New Jersey in a central engineering group. And then my second one was in Elkton, Virginia in a more technical role. The other nice thing about these is that I had been doing my work in Michigan. I was able to travel to New Jersey and to Virginia, learn some new areas of the country, and also interact with a lot of great people. So I would highly recommend internships if those are indeed available to you. So my internships really led me into my first real job. Again, this was at Merck. this time in a third location, so in West Point, Pennsylvania, where I was a staff chemical engineer in pharmaceutical R&D. So as I mentioned, how did I get this? It was from my previous internships, and I was actually able to get a role before I graduated. That just allowed me to kind of know what I was doing and plan for the future after graduation. In terms of my responsibilities, I was responsible for scaling up processes. So these are processes used to manufacture small molecule solid dosage forms like, say, granulation, tableting, media milling, and then also transferring this process to larger scales into the commercial development plant. Along with this, there came a lot of development documentation. So when you're developing a drug candidate, you have to document everything you're doing, and of course, relay those not only to your partners within your company, but certainly with the regulatory agencies as well. So that was an important part of my job. And then I also spent some time manufacturing clinical supplies, which really taught me about good manufacturing processes and how to make sure that the supplies we were making were safe and efficacious for the patient. What I really wanted to highlight from this role is some of the skills that were really important. So the first thing I noticed was the importance of that multidisciplinary collaboration. So a lot of the time when you're in a role like this, you're working with people on a project team that makes up, for example, quality assurance, analytical development, pre-formulation, all these different parts of the organization that have a stake in making sure a drug candidate gets to market. So it's really important to have the skills to collaborate with those people, understand that while everyone has the common goal of getting the drug to market, they all have their own priorities. And so it's working to make sure that everyone is able to get their job done and do it collaboratively. So you're never sitting there in your office or in the lab working on your own. That almost never happens in industry, I would say. The other thing is prioritization. So I had to juggle things that needed to be done on a day-to-day or weekly basis, for example, like manufacturing clinical supplies, with things that had longer term deadlines. So I had to contribute to writing a biobatch report, which was an important part of the documentation that was needed to the FDA. So I really had to plan for that and prioritize. It seems like something like, okay, that's not due for a few months, but if you never start working on it, then the deadline will sneak up on you. So I found those to be a couple of skills that were really important. The other thing I learned in this role is just really the impact of the colleagues around you. So I was fortunate to be working with colleagues that were a lot of fun to work with. We had lunch together. We even spent time outside of work. They were also very positive and really interested in learning. So I came from a chemical engineering background, and what really attracted me to this role was during the interview process, they were talking about how interesting it was to apply chemical engineering principles to pharmaceutical process development and manufacturing. To me, that was really exciting because when I was in college, we were applying a lot of our principles to the auto industry, the petroleum industry, and things like that. So I hadn't really thought about how you could apply it to pharma, and so I found that to be really exciting. The other thing that was important was I realized that I was asked to have some flexible working hours. So because I was working in process development and commercialization, there were times where I was asked to come in the evening, on the weekends. There was even a period of time where I was asked to switch my shift from the 8 to 5 more to the like noon to 8 type of period. So that was something else that might be important to understand when you're going into a role and what might be expected. The other thing that I really liked about Merck was the external engagement. So my department was constantly bringing in academic professors and other experts out there in the industry that we could learn from and collaborate with. For example, we were working with a professor that was studying the physics behind particulate flow. And so it was a really theoretical discipline, but we were able to really apply it to the processes that we were doing. For example, when a powder is flowing through a tablet press that you're then going to make into that final dosage form to give to patients. So that was something that I also found engaging about this particular role. Um, so I did shift companies and roles just despite my enjoyment at Merck. I decided to make a switch to Pfizer in Ann Arbor, Michigan. It was a similar type of role in terms of being a scientist and also an engineer in the drug product space. But the difference was in my Merck role, I was working in the later stages, say phase 2 through commercial, whereas in this role, it was working in pre-formulation as well as first in human and phase 1 clinical supplies. What got me into this role was my peer network. So I had been working with someone at Merck who had made the switch to Pfizer and was an advocate for me making the switch as well. The other nice thing was that this put me in a geographical area close to my family and friends. So that was a reason I made the switch, which is often a reason for people, right? It's that work-life balance and making sure that you have that support system outside of work that can be important. In this role, I did some clinical batch coordination. So that was making sure that if we were going to make a new batch of clinical supplies, say for phase 1, that we had the schedule, the raw materials, the documentation, the personnel, everything that was needed to make sure that went smoothly. I also did some new drug candidate process optimization and also some novel equipment implementation. And I'll get into these a little bit more in a second and why they were really rewarding parts of the job. So the key skills here, again, planning. So in coordinating a clinical batch getting made, you really don't want anything to go wrong. Your colleagues in the clinical space have painstakingly planned these clinical trials, and you want to make sure that you're not the person that's having us miss that schedule because you didn't get the supplies made in time. So super critical to be planning. Also problem solving. So even though all or most of us have a technical background, there's just so many soft skills that are important in roles like this. So I had problem solving I needed to do in terms of personnel. So I might have manufacturing technicians manufacturing a batch. Maybe they're not getting along with each other, or maybe they have some other things that are stopping them from manufacturing things on time. And so it can get really interesting and also just rewarding and helping you to expand that skill set from the technical into the soft skills. The other thing, I had to do problem solving in this new product optimization. So in many cases, this is the first time we were actually making these supplies at the scale we were making them. So inevitably, things are going to go wrong that you have to learn how to deal with. And then also critical thinking. So one of the things I did was I brought a new piece of equipment into the pilot plant and into the clinical manufacturing space. To do that, you really have to anticipate what can go wrong because you don't want to bring in a new piece of equipment and have all these things happen that you weren't necessarily expecting or planning for. So those 3 skills were really important. The other thing was some additional learnings from this role were really how much I thrived on leadership and autonomy. So especially through that novel equipment implementation, even though I was a junior engineer, I was really the one leading the project, deciding how to go about it, and also being held accountable for whether it went well or not. I found that to be really rewarding and really made me want to go to work every day, knowing that they were willing to take that risk on me and also give me that opportunity. The other thing I liked from that opportunity was the ability to teach. So I was able to go around to different global sites within Pfizer, teach them how the equipment works, how to develop those workflows. And I found that really rewarding as well. And I mentioned that global reach in terms of the key learnings was something that I found to be really important. And just to contrast that a little bit with some other roles, this allowed me to meet new people, see how Pfizer works in other sites in other countries. Just gave me a really nice perspective into things. You might find yourself in a role where maybe the company only has one site, or maybe you're just asked to focus on something more narrow within your site, and you might love that. That might be what motivates you. But that's just one way to kind of compare and contrast how the role might go. So I had learned a lot from this role, and I decided to stay within Pfizer, also in Ann Arbor, Michigan. But this time I moved on to a department called Research Formulations. This group was really in charge of the biopharmaceutics and the preclinical assessment of new drug candidates. And the way I got this role was from interacting with the research formulations manager. So while I was working in the previous role, I got to take a training course from this manager, also troubleshoot some issues with this manager. This particular person was really strong in physical and mechanical property analysis of solid dosage forms. So we brought him and his team in to help us with some issues we were having. So because we had those good interactions, he saw some potential in me. When he had a position become available, then he recruited me for that position. So that's a very common way that people get movement throughout a company and introduction into new roles. So with this role, this brought me a little bit out of that engineering and process development space and more into, I would say, the scientific space, and eventually moved me much closer to what I would be doing in graduate school. So this was really thinking about the physical and mechanical properties of drug substances and excipients, how they come together to make a good drug product formulation. And in doing that, that formulation development piece and that analysis piece allowed me a seat at the table on that project team, helping to make recommendations on which formulation to move forward with. And so in order to do that effectively, I really needed to be good at data analysis and interpretation, experimental skills. And so these are things pretty closely aligned with what is done in graduate school. So no surprises there in terms of what is going to be effective in that type of role. The other thing was confidence, though. So I had to be comfortable enough with my abilities to go to the project team and tell them what formulation approach I think should be taken. I think that can be hard for a lot of people, especially when they're young. They don't have a lot of experience. They don't want to put themselves out there and make that recommendation and, ultimately have the formulation fail. But I think that's a really important skill to develop as you become comfortable. And then really that's going to project you throughout your career to just get the roles you want and just thrive in the type of environment that's expected. So I think that's really important, and the more and more you can build that throughout graduate school, the better. One thing I learned here was I realized the diversity in roles and knowledge that come within the pharmaceutical space. So I had spent a lot of my career thinking about solid dosage forms, process development. I really didn't understand that much about, okay, once the patient actually takes this medication, what happens? How is the body going to interact with the drug, and how is the drug going to interact with the body? And that was something I was finding myself really wanting to learn more about. What I realized, though, was being at a large pharmaceutical company within a specific role, there wasn't a lot of time for me to delve into those questions. So I was really expected to spend, most of my time on my day job, being really good at physical and chemical property analysis and formulation, decision making. So, I wasn't given a lot of time to be thinking about the physiology and the drug interactions with the body, unless I wanted to do that on my downtime, say nights and weekends. So that's one reason that I eventually went into grad school, and there was another reason I'll go into in a second as well. The other thing that this taught me was because there's so high compound attrition, right? There's um, you're in the pre-formulation space, you're screening a lot of candidates, but only a small fraction of those are actually going to make it into late stage clinical trials, and very, very small fraction is actually going to make that to the market. So companies really design workflows and efficiencies in screening candidates. What that can do is save money, make things more efficient, but it can also maybe for curious scientists make things a little less interesting because you're essentially taking a candidate through a process and not necessarily taking a lot of time to understand the intricacies of that particular drug candidate. So those were some things I learned. And in this role, I had been thinking about going back to grad school, but what really prompted me to do so was Pfizer decided to close the site in Ann Arbor, Michigan. That left me without a job in Michigan. I had, alternatives to move other places within Pfizer, but it was just a good chance to say, okay, well, I think I'm going to go to grad school. But I mentioned that site closure because that's I'd say a relatively common thing that happens with companies, especially pharmaceutical companies, where there are mergers, there are closures, and it's just important to realize that you might be making some career switches without really planning to in the first place. So when I went back to academia, what I really wanted to do was make sure that my PhD work was going to be relevant and applicable and applicable to jumping back into industry. So the whole time I was planning on going back into industry. So I really wanted to make sure that I wasn't doing something super theoretical, but I was maintaining a nice balance between theoretical and practical. So this allowed me to expand my knowledge into the in vivo oral bioperformance space. But as I said, I was able to perform research with practical significance to the industry. So what I actually did was help to develop in vitro testing methods that were biopredictive or biorelevant. So these were in vitro methods that would allow us to ascertain whether we thought the drug product was actually going to perform well in vivo, that is, reach the plasma concentration time profiles that we were expecting and wanting. One really good thing I got out of my PhD and postdoc work was some cross-institutional collaborations. So I got to work with people in other countries at companies. So my research was actually funded by AbbVie, which allowed it to be very industry relevant. And I also continued to attend conferences and network. So another thing I'll highlight today is just how important it is to do that, how important it is to network with people. As you've already seen, networking within my companies allowed me to do different roles. And then also it will just continue even once you have that first and second role to expand your reach even further. So after my PhD and postdoc work, I rejoined industry. This time I was at a contract development and manufacturing organization. So this is Lonza in Bend, Oregon. It started out as Bend Research and was acquired later by Capsugel and then by Lonza. So again, this was through networking. So I was attending a Gordon Research Conference in the New England area, and I was talking with someone from Lonza, talking about my research that I was doing, and that really landed me the interview and then ultimately getting the position at Lonza. One thing I'll highlight later as well is, using my elevator pitch about what my research was about and how it was impactful allowed me to really get that initial interview. So I was actually having dinner with the hiring manager. I gave my elevator pitch, and they were like, "Oh, that sounds really interesting. Let's move forward with an interview." So in terms of my responsibilities, I was leading teams to develop new technologies and technical services, and also driving R&D strategy. And so in this role, I was doing even more continued communication and collaboration with others. But because the position I was in, I really had to understand how to communicate with non-scientists. So in graduate school, in our work, we're very, let's say, invested in the technical details. We have to think about all those details to really make our experiments run smoothly. But then when we go to talk with the person that's funding our research, so in the case of my role, we had to get the head of our division to give us money to do our research. I had to explain why this research was impactful, how it would help our business, and ultimately how I think I could get it done and, in how much time. So that person didn't want to talk about the intricate details of my experiments, but really wanted the high level details about what it was going to achieve in the end. So even though that was something that I did a little bit later in my career, I think it's important the earlier you can start, the better in terms of being able to convey those details. I think one good way to start is just, tell your parents, your friends, what you're doing. Many of those people are not going to be scientists, so see if you can communicate what you're doing with them and get their feedback in terms of their understanding and also their opinions into how impactful they think that research sounds to them. The other thing that was important here was coaching. So I was actually leading technical teams of junior level scientists and engineers. So I need to understand to coach them to get things done and to further their careers as well. Some additional key learnings were educating others. I'll get into this in a little bit later as well, but when you're working at a CDMO, oftentimes you're educating clients. So these are the people that are bringing their molecules to you. That can be really rewarding because oftentimes these people have no insight or experience in your area. And so you really have to start from scratch and educate them about what you're doing and why you're doing it. The other thing was marketing. So at CDMOs, a lot of the time you have to market the CDMO, you have to market the technical expertise, for example, in your particular role. You want clients to come to you with their molecules. So oftentimes I'd find myself doing external webinars, external presentations. That can be really rewarding and can also help your career. But that's a little bit divergent, might take you outside your comfort zone in terms of if you're someone that likes to be in the lab doing science only, you might be a little bit uncomfortable getting out there and projecting yourself, but it's something that could be expected of you in a CDMO type role. Other things I had to think about were how to govern projects, how to create new project ideas. And these are just really about setting milestones throughout your project journey to understand if your project is on track, if you need to change strategy, or if you need to stop the project because it's not working and then move on to something else. This might resonate with some of you even in your PhD research. I'm sure many of you have guidance from your thesis committees in terms of making sure that you have those intermittent milestones so that you're not spending, say, 3 years off track and finding yourself then needing to start over. Okay, so my last summary of a role, from the CDMO Lonza, I then pivoted just in the last few months into modeling and simulations with Simulations Plus. As Suvrachala mentioned, I'm a PBPK R&D scientist. And what really brought me into this role was the fact that I was using this software in my previous role. So at Lonza, we were using Gastro Plus and AdMet Predictor, which are two of Simulations Plus software packages to predict the in vivo exposure of our drug candidates in animals and in humans. And so I learned how to use the software, and I also got to interact with Simulations Plus colleagues. So this really got me into the next role and let me do something, not that I wasn't necessarily trained in, but it was adjacent to my training. So I have a little bit of experience developing software code, for example, but not a lot. And so being able to, I'll say, dabble or even, more strongly get trained in the software in one role allowed me to gain that other role. And I find that's another way that you can get into roles that don't necessarily align with your training, is to have that opportunity to start getting exposure to something new in your current role. So with this, I'm developing mechanistic software features in GastroPlus, making it better, making it more aligned with physiological considerations as the research evolves and we get more data allowing us to build these new features. I also get to support research grants and also provide training. Key skills here, it's that critical thinking. So when doing PBPK simulations, we really need to think critically about what our input parameters are, what the software is doing, and understanding whether or not we think that aligns with how the drug is performing in vivo. Also adaptability, so going into a new area, going into software um, versus being someone that's in the lab doing in vitro experiments has just caused me to adapt a little bit to that change. Another reason for me doing this role is I really wanted to expand my learning. So I had done a lot of roles within the absorption side of things. So how does the drug absorb in the body? But in moving into this new role, I can start to learn other areas like how the drug distributes, is metabolized, and is excreted from the body. So I think it's really good throughout your career to evaluate how much have you learned in your current role, what is the potential for learning more, and then whether or not you want to make a change to a different company or even within your same company to learn something brand new. Okay, so I've talked about this a little bit, but I wanted to talk a little bit more about CDMO versus pharma. So when you're at a CDMO, you see a high diversity in drug candidates. So you're seeing drug candidates from multiple different companies. Depending on the CDMO, you might have 10 to 50 companies you're working with. And so they're all working in different therapeutic areas. They might bring formulations to you that were made using their favorite technology, their favorite excipients. So by working with all those different candidates, you can really learn a lot. Um, in contrast, when you're at a pharmaceutical company, maybe you're working in a single therapeutic area. Maybe your company, because of those efficiencies that are needed when developing candidates, they might use a platform formulation. They might try to use, a very similar formulation from one candidate to the next, just to get a readout in the clinic. So your experience might be a little bit more limited to those formulation types and process types. As I'm, going through this slide, these things aren't universally true, but just trying to make some high-level observations from my experience. The other thing that I've mentioned is CDMOs are service-oriented, so they care about the product, but really what they're trying to do is execute the service that the client is asking for. With that comes the need to interact with clients a lot, make sure that you're making them happy, make sure that you're executing those tasks that they asked for. versus with being product oriented, at the end of the day, what you need to do is make sure that you're meeting all the objectives within your company to get that product to market. With that, in the service CDMO side of things, you're routinely externally facing. So if you're working directly with clients, you're meeting with those different clients throughout the week. You might be meeting with clients that are on the other side of the globe. You might have to be flexible in terms of what time you're meeting. You have to be ready. You might be balancing molecules from 2 to 5 different clients. So you have to keep all those molecules and those clients straight and really make sure that you're projecting to the client your credibility, your technical credibility, your ability to get things done. As I mentioned, that often gives the chance to teach the client about what you're doing, which can be really rewarding if that's something that you like. The other thing that comes with that, though, is sometimes the client isn't going to give you all the information that they have about their molecule. They might be hesitant for IP reasons to give you all the data. So sometimes you're working on molecules where you don't have all the pieces of the puzzle. In big pharma, you're going to have more internal meetings, and you're going to still be meeting deadlines, but again, you're going to be more internally facing, even though in some cases you're going to be working with a company that has some academic collaborations and things like that. In terms of when you're evaluating a new role, here are some things that I think are important to consider. So is the role relatively routine and workflow driven, or is it going to give you a chance for learning a diverse skill set and allowing you to be creative? I've kind of touched upon this already, but are you going to go into a role where you're in a large department within a pharmaceutical industry where you're specializing, let's say in analytical chemistry, maybe your role is running HPLC to analyze a concentration time profile within an in vitro experiment. Maybe that's what you're going to do day in and day out. Maybe that is going to be rewarding for you. That certainly has an opportunity for problem solving because all drugs are different. But some other people might not enjoy that. They might want to be in more of an R&D role where they're doing all different kinds of experiments. No one told them, what's the scope of work, what's the procedure for running this, and they have to come up with that themselves. So I've seen this make or break someone's happiness in terms of the role that they're in. Touched upon this, but is your role going to be limited, or are you going to have a breadth in your scope of work? So, maybe if you're in a large department, you're going to have a more limited skill set. Maybe if you're in a really small startup company, it's kind of all hands on deck. Everyone learns how to do everything. Maybe that's a role that you're going to thrive in more. Are there opportunities to publish and present externally if that's something that you're interested in? If there are opportunities to publish and present, is that something that you're allowed to do during the day, or are you expected to do it on nights and weekends? And even if theoretically your company says that you can do this, it's also good to check the track record of other colleagues that are already at that company and that have they been able to publish. Oftentimes it can be tough to publish in the pharma industry because companies are working on new drug candidates and they don't want that information about that to be external, especially before approval. The other thing, are there training and educational opportunities available either inside or outside the company? Is there training money set aside if you want to go do an external course? Are they going to be able to support you in that endeavor if that's important to you? Getting into a role outside of your field, focus on your educational fundamentals and how they're applicable. So even if you don't have a pharmaceutical background, I've seen people that have a physics background or a chemical engineering background with no touch, no ties to the pharmaceutical field that have really highlighted those fundamental skills and how they can be applied to the new role. I think that can be a very effective way to get yourself into a new field. Highlight your soft skills and your onboarding track record. So really highlight those skills that you learned in grad school or a previous role, and how those can be applied, and also how quickly you've been able to be trained on things. That can be really important. Use your current role as a jumping off point. So I gave some examples where I was able to work on a certain technology, a certain software in a current role, that then gave me just enough experience to show that I would be effective in the new role. Foster and project confidence. So, definitely be humble while you're doing this, but I think it can be really effective, again, to project, yes, I know my background isn't in this field. However, my educational fundamentals, my soft skills, and my onboarding track record, I think will really allow me to do a good job in the next role. In terms of resume and interviewing tips, I really think it's important to demonstrate your critical thinking skills. So I've seen PhD students come and interview for a role. They give their presentation, and then someone asks them, okay, well, why did you decide to go about the experiment that way? And unfortunately, some people will say, oh, that's just what the last person did, or that's what my professor said to do. And I think that's definitely a red flag for the hiring team. What they want to hear instead is, okay, I chose that particular way to run the experiment because I analyzed the properties of the drug, and that, caused me to select this particular way of doing things. If, it really was that your professor, told you to do it that way, find some other ways to demonstrate those critical thinking skills, or analyze the way you did it and say, hey, I did this because that's, what my professor said to do. However, when I think about it, we could have done it, also these other ways, and this is why. So just make sure that you're showing that you can think for yourself. Highlight the positive outcomes of your research or your previous work, and also be quantitative. So on your CV, you might say, I brought in a new piece of equipment to the pilot plant that allowed us to manufacture clinical supplies in 1 week versus in 1 month, or save X amount of dollars, or solve this problem where, we weren't able to manufacture a certain type of formulation before, but now we can. And then refrain from writing things like ran HPLC, ran, this process. I'd say rather than focusing on the process that you used, really focus on the positive outcomes of what you did. Practice your elevator pitch. I already mentioned this one. So at a high level, describe the background of what you're doing, the challenges in that particular field, and then what your research did to solve those challenges, and what the outcomes were. Ask questions. So definitely ask questions at your interview. If you have trouble thinking of questions before your interview, you can research the company, what they do, what processes they run, what equipment they use, what analytical techniques they use. That'll allow you to show that you've done your research and allow you to think of questions. If you still can't think of questions, you can go online and Google, good questions to ask for an interview. And I think that can be super good too, in terms of coming up with good questions. And these questions, might be more around things I've talked about, like what's the environment? Can we publish? What are working hours like? What's the training like? And, just remember that you're interviewing them as much as they're interviewing you. So to recap, some of the things I wanted to highlight, internships are very valuable if you can do that during your graduate work. If you can't, you can still rely on your research. That's one great thing about graduate school in this area is that you're definitely going to be doing lab work, and so you can rely on that for your technical hands-on experience. Network and market your science. So the networking is super critical, as I've demonstrated. Basically, every role I got was through networking. And then marketing your science in terms of writing papers, attending conferences really helps as well. And then finally, think critically about what you want in a role in terms of some of the things I mentioned, like how routine is it versus how creative is it? Are you going to be focusing on one skill or on multiple skills? What's the opportunity for having global insight into what's happening at the company versus, working in one department at one geographic location, for example. I want to acknowledge some of my PhD advisors, Greg and Gordon Amadon, as well as many of my previous managers and collaborators as well. And then with that, I think we have a decent amount of time for questions. And as I mentioned, feel free to reach out to me via email if you have questions later.
Deanna, thank you so much for a wonderful webinar. We really much appreciate your time today. So that being said, please feel free to post questions in the chat. If you feel comfortable coming off of mute, please do that as well. We are now open for questions, and we have about 10 minutes left to ask of any questions that you may have.
Hi, I may start. This is Shang Hao. I already working in industry now, but I feel that if I had this opportunity to learn in grad school, that would be super great. And you have very unique experience that you actually after graduate with a bachelor degree, then you work in industry, then you go back to PhD study, then go back to industry again. So that's very unique. And my question is, so you may touch this a little bit, but I want to ask you how you during this process when you go back to for your PhD study, how you select your research topic and how this impacts you for your future career in industry.
Yeah, so in terms of selecting my research topic, I wanted to pick personally something that I thought was relevant to my previous experience and would be relevant to my future experience. So I had spent a lot of time understanding how to manufacture solid oral dosage forms, but I want to understand the physiology behind, when a patient takes this dosage form, what is the body doing to it? So that allowed me to think in terms of biopharmaceutics, physiology, oral, drug delivery. So that led me to select that particular topic. And then within that, I think there are other considerations in grad school, right? So you might go to a particular school and there may be, 3 to 10 topics, let's say, depending on how many professors there are. I think it's also about talking to all the different professors in your school and seeing who do you fit well with, who motivates you, who excites you. Talk to the grad students in those labs as well, and then make sure you find a good fit. So it's kind of the topic as well as the advisor. And then, as I had mentioned, I also wanted something practical. So I was looking at the topic and the actual research and saying, okay, this particular professor interacts with industrial partners a lot, has some collaborations where in that case we were being funded by AbbVie. And I thought, okay, if I work on that, then I know what I'm doing is going to be something that AbbVie cares about, and so therefore is practical and isn't so far on the theoretical side of things as to not be as attractive. Does that answer your question? Or
Yes, thank
okay,
you.
sure.
So Deanna, I have a question. One of the bullet points that stuck out to me, I believe it was for your role that you took on the formulations aspect at Pfizer. Communication with non-scientists. How do you recommend working on this role for a graduate student, especially coming from someone like me who's been in industry now for about 5 years, but still struggles to communicate with non-scientists?
Yeah, so I think one of the first things is thinking about what motivates the person that you're talking to, kind of what are they looking for in the end of the day. So um, this this could this could vary. So um, in one case, I was trying to convince the head of the division to provide us money to do our research, right? So in that particular case, I know, okay, this person wants to understand how this technology we're developing is going to allow us to get more clients or save money or save time. So then I had to think about my research. And so if I give an example, one of the things I was working on was developing a new technology for making amorphous solid dispersions. And so I thought, okay, well, this technology will allow us to manufacture drug candidates that we weren't able to manufacture before with the other technology. So, okay, that opens up more candidates that we can address. Also, this technology is more environmentally friendly, so that's going to attract people, wanting to do more for the environment. Also, this technology is going to allow us to do the manufacturing in half the time. So I really focused on those points rather than focusing on the intricate details about the actual technology. Um, the other thing I would say is sometimes you're educating people. So you're educating people about what you're doing in terms of, okay, I'm using an in vitro dissolution test to understand how a drug performs in vivo. Rather than, let's say you're going to someone that's, not in science or is just in a totally different field. You wouldn't want to start out by describing, okay, well, the apparatus uses, pH 6.5 with 500 mils of media, and we stir it and we, use HPLC and all this stuff because they're just going to be like, okay, I don't understand the context. So you really want to start off with, okay, let's say an oral drugs are really important because most people prefer oral drugs. It takes up most of the drug pipeline. However, before we can develop an oral drug, we have to decide if it works. And, before we put it in people, we need to test it in the lab and, really start from that really basic information to first like pull them into what you're doing at a high level. And then, kind of talk through the details as people ask questions.
That's very helpful. Thank you. So we have a question in the chat. This is from Rodrigo. He says, "Hello, I'm working in a CRO right now, and I'm doing a master's degree in pharmacokinetics at the same time. Do you think I can migrate internally once finished with my master's degree?"
Okay, yeah, and I think you mean migrate internally into like a DMPK or really pharmacokinetic focused discipline is what I'm assuming. And I think, it depends on the company. I know someone at a company that did something similar to you, so they took a course while working. And what they did was actually during that coursework, they reached out to someone within their company that worked in that area, started shadowing that person, telling that person about what they were doing, and kind of getting an early, really exposing themselves early to those people, and then ultimately making that transition. So again, I would say it depends on the company, but I think again that networking piece is important. So, don't necessarily wait till you have your master's and then just, apply through the website or your internal, intranet to it, but really seek out the people in that other department, talk with them, interact with them, show them your interest and your new expertise, and then try to make things happen that way. It might not happen right away if there's not an opening, but if they know that you're someone that's motivated and trained in that area, then, maybe 6 months, a year down the line, an opportunity could present itself.
Sheng Hao, I see you posted a question if you'd like to ask it.
Yeah, because I saw there would be no time to ask. That's why I typed. Anyway, so my question is, um, you are now working in a software company. It's a simulation plus. It seems that you are developing software for people to develop the drugs to help them to collect the properties or of course based on sometimes based on experiment data, sometimes you have to based on the publications, sometimes it is purely on theoretical prediction. So my question is, how, in your opinion, how AI could help this field for drug development at the preclinical stage, for example, let's say specific for drug-like properties or other properties. I just want to be specific, so you can like define your topic a little bit more. Yeah, that's my question. What are the
Okay, yeah.
unique challenges in this field when you work for this company?
Yeah, I'd love to put Suvrachala on the spot for this one since she's been working at my company longer, but I'll start by saying one thing that we do, of course, is the drug property prediction with ADMET predictor. So I find it really helpful to be able to predict drug properties, right, without measuring them. This also resonates with my work at Lonza, where we were trying to build these models and we didn't necessarily have all the data. We at Simulations Plus are using the ADMET predictor software to do these property predictions early on and continuing to feed more data in, make better and better predictions. And I really find that to be instrumental, especially when you have a large amount of compounds that you're trying to analyze. So I don't know if I have anything really specific for you other than that. It's important. I think we're always trying to make further and further gains along those lines, and I think it's going to be more and more a part of the way things are done as we move in the future. And if Suvrachala wants to add anything, I invite her to do that.
Yeah, Deanna. So what happens is by using the admit predictor, we really know even before we get a lot of information from the client, we get to understand the classification, like what's the possible clearance pathway is, what are the enzymes involved. So that gives us a basic idea to start developing the model. So most of the times, um, the client can provide us with some of the properties, but in some scenarios, the in vitro assay may not be reliable, or sometimes the assays are not done. So in that case, the admin predictor that we have is really helpful to understand the basic behavior or the drug-like properties of the molecule.
Thank you. Thank both of you for your insights information. Thank you.
So with that, we are at time for this webinar. I want to say again, a big thank you to Deanna and spending time talking through her career. The bouncing between of academia and industry is definitely one that is an awesome perspective to share. As was stated at the beginning, this was recorded. I will share this via our AAPS PDPD community library that we have access to via aaps.org. I will also post in that posting chat the email for Deanna, so anyone who is interested in reaching out to her, you may do so. Again, thanks everybody, and have a great rest of your day.
Bye. Thank you.